Hi students, very good morning all. Welcome back to Sri Ramanuja Vidya Mandir. Today I am going to explain about the reproduction in the plants. Sexual reproduction in the case of the plants. So as we all know that flower is the reproductive part of plant. Here our plant is having n number of different parts such as roots, stems, leaves, flowers, fruits and all. Out of all this, flower is considered as the reproductive unit of plant. Understand? Now, I think already I have discussed all these in the last class. Once again, I would like to give a small review of the previous class. Then I'll start today's topic. See students, structure of flower. Actually, a typical structure of flower consists of 4 different parts. Those are sepals, petals, stamen and the carpels. Sepals, petals, and the center we have the stamen and the carpel. So sepals, these are the first wall of the flower. The main purpose of the sepals is the protection. They're going to offer protection to the internal parts of the flower. So petals, so they'll be colored. So due to this good color and attractive color, so they'll be attract the insects for the purpose of the pollination. Next, stamen. See, first two are like sepals and the petals are the non-reproductive units of the flower. So whereas stamens and the carpels, they are the reproductive units of the flower. So stamen is nothing but the female reproductive unit. This stamen consists of 2 important parts, filament and the anther. So anther is the region where we can see the production of male gametes. Okay, they are nothing but the pollen grains. Let us see this last one. This is the carpel, and the swollen part of the ovary at the base. It is present, and a middle one is called style, and the head portion is called stigma. The ovary contains the ovules, and each ovule possesses an egg, which is this egg is nothing but the female gametes. Right. So structure of anther. Actually structure of anther is bilobed, and we can also call this as this particular position dithecus, and a longitudinal groove separates the theca. And see students, this this later if you'll examine the cross section of the anther, actually this one. So see students, also the next diagram. So now this is the cross section of the anther, and so if you'll see the structure of anther means we can observe the presence of 4 different layers. Okay, first one is epidermis. Next to this endothecious layer is there, and middle layer, and the 4th one is tapetum. Out of all 4, first 3 they'll be offering protection to the anther. And let us talk about the tapetum. See, tapetum is having the here tapetum is innermost layered. It is having the cells of diploid or polyploid condition, having binucleated and multinucleated cells. This tapetum nourishes the developing microscope or pollen grain, while the other three layers of the cells, they'll be giving the protection to the developing anther. And at maturity, 2 microsporangios of each lobe becomes fused to form sac. So initially they'll be looking like tetrad, but when it is matured, how it looks students? At maturity, the 2 microsporangia of each lobe becomes fused to form pollen sac. See students, this one is the pollen sac. When it is on rupture, they are going to release the pollen grains. That particular breakdown of the anther to release the pollen grains is known as dehiscence, we'll call. Right. And microsporangium and the formation of the pollen grain. So let us discuss how the pollen grains are synthesized or formed in the anther. Like a typical microsporangium appears. So this is the microsporangium, students. So how it will appear? So it appears circular in outline, but it is surrounded by the 4 layers of cell. In the before slide only I have discussed. There are epidermis, endothelium, and the middle layer, and the final layer is tapetum. So as you all know that the outer 3 layers perform the function of protection and helps in the dehiscence of anther. Dehiscence means breakdown of the anther to release the pollen grains out. So the innermost layer, as it is tapetum, so which is going to nourish, means which is going to provide the nutrients, essential factors for developing pollen grain. Right. So microsporogenesis, production of microspore, the process of formation of microspore is microsporogenesis. Right. So when the anther is young, a group of compactly arranged homogeneous cells are present. They'll be located at the center, which occupies the position of center of the each microsporangia. So this is the microsporangia. So as at the center it is having the homogeneous cells. As the anther develops, so the cells of sporangium tissue undergoes meiotic divisions to form microspore tetrad. So students, when it is developing, so this sporogenous tissue will develop to form a microspore tetrad. See, and each sporogenous cells capable of forming microspore. So microspore tetrad, each sporogenous cell, students listen, each cell have the capability of forming tetrad. And see, and this is called pollen mother cells, means the initially the cell which is going to convert into the tetrad. So that initial cell is known as PMC, pollen mother cell. So the process of formation of microspore from the PMC through the meiosis is called microsporogenesis. By what division, students? It will get a microspores. By the process of meiotic division, we are going to get the microspores. That process is known as microsporogenesis. So students, I am repeating each and every point. Okay, so why? Because it is the connecting topic. Okay, so you need to, first of all, you should have an idea about the proper structure of anther, pollen grain, male reproductive part of the flower. If you'll have the correct idea about the structure, obviously you can remember all the developmental stages very easily. Right. So as the anther matures and dehydrates, microspores dissociate from each other and develop into pollen grains. Each microsporangium has several thousands of microspores or the pollen grains that are released with the dehiscence of anther. See students, each microsporangium have thousands of microspores. They are nothing but the pollen grains. So they will be released out when the dehiscence of anther happens, means when the anther breaks down to release the pollen grains. Right, now let us see the structure of pollen grain. Pollen grain is nothing but the male gamete. Okay, microspore. So pollen grains represent the male gametophyte. See students, so this pollen grains represents the male gametophyte. Okay. Next, pollen grains are generally spherical, measures about 25 to 50 micrometers in diameter. It is a size, students. It is in between 25 to 50 micrometers in diameter. It has 2 layers of the walls. See, this pollen grain is surrounded by the 2 layers of the walls. The outer one is hard layer, is known as the outer layer is called exine. So which is hard in nature, which is made up of one of the most resistant organic material known as sporopollenin. See students, why it is so hard means, so it is made up of material known as sporopollenin. Okay, it can withstand the high temperatures, strong acids, alkali and the enzymes. Why it is so hard in nature means, as it is having the special structural material known as sporopollenin, so which has the capability of the withstanding, see, the high temperatures, strong acids, alkali and the enzymes. The exin has apertures called germ pores. See students, here I said no, this is the outer layer. This outer layer is known as exin. What is this students? Exin. It is very hard in appearance. So why it is so hard? Due to the presence of a special structural material known as sporopollenin. Okay, and reason is that so the exin has the aperture, means it is having a small fine gaps, and that aperture is nothing but the students germ pore. What is that? Germ pore. Right? Now see, where the sporopollenin is absent, this exin has aperture, here there is no presence of sporopollenin. So as it is having the gap hole, it doesn't have the what? Sporopollenin substance. Right. Again, the exin has various patterns and the designs which helps in the identification. It is having very peculiar patterns and design. So by observing these patterns and the design, we could easily identify the, as it is, exin of pollen grain. And so the inner wall of the pollen grain is known as intine. So students, we discussed about the outer layer. Now inner layer of the pollen grain is known as, students, what is that? Intine. Okay, so this, it is thin continuous layer made up of cellulose and the pectin. So this layer is made up of, what are the pigment substances students? Cellulose and the pectin. Okay, so inner one is known as intine. So students, this is the inner layer, intine, made up of substances known as pectin and the cellulose. Cytoplasm of pollen grain is surrounded by the plasma membrane. See, at the center it is filled with the cytoplasm. Okay, and this cytoplasm is surrounded by the plasma membrane. Next to this, it is having intine and the exine. And see, a mature pollen grain contains two cells, vegetative cells and the generative cells. So actually, when the pollen grains matured, so it is going to contain two different types of cells. First one is generative cells and the vegetative nucleus. This red color circle indicates the vegetative nucleus, and the second one is, it's like eye-like thing is there, no center, that is the generative cell. So this is the structural description about the pollen grain. So once again I am telling, pollen grain is also known as gametophyte. Where it is produced, students? It is produced in the anther, the microsporangium of the anther, right? And this, it is having the diameter of 25 to 50 micrometers in diameter. It is surrounded by the two different layers. Outer one is called exin and the inner one is called intine. Okay, outer layer is so hard in nature. The reason is that it is having a special material known as sporopollenin. Okay, it is one of the protein. Due to the presence of this special protein, this exin can withstand the high temperatures, high acids and alkalis and the enzymes. And so it is, and the inner layer is known as intine. It is so smooth when compared to the outer one. So it is constructed by the cellulose as well as the pectin. These are the structural components of our intine. and our pollen grain is made up of cytoplasm. This cytoplasm is surrounded by the plasma membrane. Okay, and when it is matured, means when the pollen grain gets matured, so it is having the two types of cells, generative cell and the vegetative nucleus. Understand? Let us see, students. So vegetative cell, generative cell. Actually, let us describe about the what is vegetative and what is generative. So vegetative cell is bigger, has an abundant food reserve and large irregular shaped nucleus. So students, so it is having the reserved materials, having the nucleus, okay, and generative cells are small and floats in the cytoplasm of the vegetative cells. Understand, students? They will be floated. Generative cells are little bit smaller in size, where they will be floating in the cytoplasm of the vegetative cells. So it is spindle shaped with a dense cytoplasm and it is having the small nucleus. In most of the angiosperms, pollen grains are shed at two-celled stage. In some species, generative nucleus divides mitotically to give rise to the two female gametes before pollen grains are shed. So just understand this concept, students. In some species, generative nucleus divides mitotically by means of mitotic division. It divides to give rise to the two male gametes. One generative cell nucleus is going to give rise to the two male gametes by means of mitotic division. So before the pollen grains are shed. So once the pollen grains are shed, they have to land on the stigma. Shed means what, students? So once the pollen grains are released, or which are going to come out of the anther, means, so what the immediate step they have to do? They need to land on the stigma, right, for the process of fertilization. So see, so they need to land on the stigma before lose their viability. Actually, it's a living thing, no, it has some viability, it has some period. So within that period only it has to reach the stigma. Okay, if have to bring about the fertilization, within hours, so the pollen grain which is released from the anther should reach the stigma. Okay, the period of viability of the pollen grains varies from 30 minutes to the few months. It depends upon the species. So it varies from 30 minutes to the few months. So ovule is small structure attached at the placenta by means of funicle. See, students, and today we are going to explain about the female gametophyte. Okay, like, see, this ovule is a small structure attached to the placenta by means of funicle. The body of ovule fused with the funicle with a region called hilum. So the hilum represents the junction between the ovule and the funicle. So I'll with the diagram I'll explain to students. See, students, this is the structure of ovule, and before this I should introduce the female reproductive unit. So far we discussed male reproductive unit, that is male gametophyte, also known as pollen grain, how it is produced, where it is produced, and we discussed. Now we came to the female reproductive unit. So gynoecium, or also known as pistil, so represents the female reproductive unit of flower. So actually, students, you just have a look on the flower, and this particular part is nothing but the pistil, or also known as gynoecium. Okay, right. So this is the pistil. What is this, students? Pistil. Okay, so female reproductive unit of the flower. Pistil means female reproductive unit of flower. So it may consist of one carpel, so they'll be called as monocarpellary. More than one carpel means bicarpellary if it is having 2 carpels, bicarpellary if it is tricarpellary, or if it is having more means multicarpellary, depending upon the number of carpels present. So we'll call this as a monocarpellary, bicarpellary, tertiary carpellary or tricarpellary, and the multicarpellary. When the number of carpels is more than 1, the gynoeciums may be syncarpous. So all the carpels are fused together. So all the carpels they'll be fused together. We'll call such condition is called syncarpous. Right? And so they are apocarpous. Carpels remains free. So if the carpels are united, means we'll call syncarpous. If the carpels are remain free, means we'll call them as apocarpous. Each pistil, this pistil has 3 main parts. What are the students? First one is main parts I'm representing now, ovary. Second part is style. Third part is students stigma. Ovary, style and stigma. Right. So let us talk about the ovary now. Ovary, the basal swollen part where the ovules are produced. See, it's the base. The swollen part is nothing but the ovary. Inside the ovary, it is having the what is students? Ovules. And see the style, elongated filamentous part. So elongated filamentous part is called style. And the next one is stigma. It is the distal part. So it acts as a platform for the landing of the pollen grain. So we can say that the head portion of this pistil is nothing but the stigma. So it will be acts as a landing platform for the landing of the pollen grains. This ovaries encloses one or more cavities called locules. Actually, this ovary is having some cavities known as locules. Okay, inside. Placenta is the tissue seen in the locule that bears ovules. So inside the locule, it is having the special tissue known as placenta. Placenta. Okay, right. And that bears ovules, known as, they are nothing but the megasporangia. So see students, if it is female gamete, microspore or microspore will call. If it is female, megasporangia. Okay. See, this bears ovules. Next, the number of ovules is an ovary may be 1 in mango, rice, wheat, or in many in the orchids, watermelon, papaya. So the number of ovules differs. In case of mango, it is 1. Rice, it is 1. Wheat, it is 1. Okay, there are some fruits, they'll be having the more. So example, watermelon and papaya. So this is the structure of pistil. So stigma, style, ovary, these are the major parts of the pistil, right? Ovule. Ovule arises as primordium on the placenta. See students, this is the structure of ovule. It is attached to the placenta by a short stalk called funicle or funiculus. Okay, so it is attached by a stalk-like thing known as funiculus, or a singular means we will call funicle. And the junction between the funicle and the ovule is called hilum. See student, this is the ovule and this is the funicle. The junction between this is called, students, what is that? Hilum. Next, and which remains as a scar on the seed. So this hilum remains as a scar or a spot on the seed. The primordium grows into a mass of cells forming nucellus body of the ovule. So this primordium goes and develop into tissues known as, so what is that? Nucellus or body of the ovule. See students, this is the whole body of the ovule. Next, nucellus becomes surrounded by two protecting envelopes called integuments. So this entire nucellum is surrounded by the protecting envelopes known as integuments. Outer integument and inner integuments. Okay, outer and inner. Okay, so except at the tip, leaving a small opening called micropyle. So they'll be this integuments is having the gap at the tip. So that tip is known as micropyle. So all these two are the integuments, inner one and outer one. So at the tip it is having a small opening. That opening is nothing but the micropyle. What is that, students? Micropyle. Okay, the basal part of the ovule is called chalaja. So this is the basal part of the ovule, is known as chalaja. Understand? Next, see, and that lies just opposite to the micropyle. So just opposite. So opposite to this, what we have? Chalaja. And the cells of nucellus are rich in reserved food materials. So they'll be having the food material. So they are going to nourish the entire ovule. And there is generally a single micro embryo sac, or also known as female gametophyte, located in the nucellus. It has developed from the megaspore. So it is the nucellus. So it is having at the center it is having the embryo sac, also called as female gametophyte, just located in the nucellus is the tissues, and it develops into megaspore. So students, so here I have explained the ovule structure, and in the now it is megasporogenesis. So the process of formation of megaspore from the megaspore mother cell is known as, what is that, students? Megasporogenesis. The process of the formation of megaspore okay, from MMC. MMC means megaspore mother cell. Megaspore mother cell. So this whole process is known as megasporogenesis. Okay, now a single MMC difference in the nucleus towards the micropylar region. So it is a large cell containing dense cytoplasm and the prominent nucleus. So MMC undergoes meiosis, results in the production of megaspore. Here our megaspore mother cell, on meiotic division, it is going to produce the 4 megaspores. So this is the megasporogenesis. So before we discussed about the microsporogenesis. So in this way we will be getting female gametophyte and the male gametophyte. That's all for today's class. Okay, let us continue the same in the next class. Okay students, thank you all.